Okay, everyone. So it's a very sad occasion because we're almost at the end of what has been a great meeting, but we have one last really exciting session to go, and it's our final keynote presenter. And I think Steve Horvath really doesn't need introduction to anyone in this audience, so I'm not going to spend ages giving him an introduction because I think we want to listen to what he has to say. But I think, he's clearly known for really making one of the most seminal discoveries, I think, in this field that, as we've heard from presentations in this meeting, have really driven a lot of the work that many of us are doing, looking at how we can predict things like accelerated aging from DNA methylation data. So I'm going to shut up and hand over to Steve.
Thank you.
And the other thing I did want to add is we're obviously delighted in the UK to have Steve over here in the UK, so it's brilliant
to have you. Thank
in.
You so much. Yeah. Thank you, John. Thank you, John, Laura, Charlotte, for organizing this amazing meeting. Thank you for giving me the opportunity to present some work. I lived the last 22 years, I think, in California, and now I'm moved to the land of opportunity, which is Britain. And I'm very happy here. I will describe our latest results from the Mammalian Methylation Consortium. So we have methylation data from hundreds of species. And I will touch on many different topics, but one of the things I want to discuss is this graphic that describes how the rate of change in methylation on the y-axis relates to 1/life, maximum lifespan of a species. So every dot is a species, and there's this rate of change in methylation. And there's a lot to be said about this equation, but anyways, so that's why I called it fundamental equations. And then this word methylation dynamics relates to rate of change. And also the previous speaker, Rosa, mentioned the importance of longitudinal studies. So fundamentally, we are looking at longitudinal studies as well in different species. And the Mammalian Methylation Consortium was really motivated by one of the axioms that exist for molecular biomarkers of aging. And that axiom is that a good biomarker of aging should apply to all mammalian species because all mammals experience aging. So wouldn't it be nice if methylation clocks could measure aging in all mammals? And here's my acknowledgement slide. The Mammalian Methylation Consortium was comprised of almost 200 researchers, many from the UK, many from Europe. And through this tremendous, so people had freezers full of tissues and DNA collected over decades. And so in total, we managed to generate methylation array data from 15,000 tissue samples from 348 different species. And the experts in the room know perhaps that the mammalian species can be clustered into 26 taxonomic orders, also or phylogenetic orders, things like primates, carnivores, bats, rodents. There are 26 taxonomic orders, and we only missed one of the 26, and this is this bizarre marsupial mole that lives somewhere in Australia, and for the life of it, we couldn't find it. And there's an opportunity for you to really make your mark in the history of science. Get a sample from this marsupial mole, And in any event, so the data are described in two papers, one by Armin Hagani, one by Ake Lu, who sits here and presented a poster. But what I'm very proud of is that we've publicly released the data. They're entirely free. There is no material transfer agreement, no legalese, no nothing. You can right-click it and download the data on your computer during this talk. And the data are, in my opinion, priceless because they really build on decades of people collecting very precious tissue samples, from hundreds of species. The data are also very clean, I want to say. And the reason why I can say it is because I was the data manager, Many professors hire an undergraduate to manage the data, but not here. So whatever data entry error you find, that's my error. But so we have 70 tissue types. Earlier today, we heard wonderful presentations on the placenta. We have placental samples from a couple of species. We have so many different cell types, rare cell types. And anyway, please delve into this data. I now want to talk about the technology that was used to generate the data. So as everyone knows, the human infinium arrays from Illumina, like the EPIC or 450, they cannot be applied to all these hundreds of mammalian species, right? And we thought that maybe one can design an array that looks at highly conserved stretches of DNA, ultra-conserved stretches, and focus on those. And this then idea was implemented by my UCLA colleague, Jason Ernst, who helped to design the so-called mammalian methylation array. And I just want to tell you what is so remarkable about it, because here you see in orange the human CPG. We want to profile it. And then you see the neighboring sequences in different species, and yellow denotes mutations in these species. But the mammalian array can tolerate some of these mutations. And therefore, then it focuses on these highly conserved regions. And but this comes at a cost. If you look at, if you focus on ultra-conserved stretches of DNA, what does it mean? You will end up with a few 10,000, not with a million, And here I show you different taxonomic orders or phylogenetic orders. Remember, there are 26 of those. Now, for primates, we have about 36,000 cytosines on the array that apply to all primate species. And then at the bottom, you see monotremata. These are these egg-laying mammals. By the way, we do have methylation data from egg-laying mammals like platypus and others. But also you see this diprotodontia, which are really marsupials. And according to that, we have, let's say, only 17,000 cytosines that apply to marsupials. But here I show you a proof of concept that the data is still exciting, because we built epigenetic clocks for all marsupials, for kangaroos, koala bear, and so on. So no problem, the array applies. But now one could do something risky. One could try to apply a mammalian array to an amphibian, right? And we have done that. And for example, we analyzed data from this clawed frog, which is a very important animal. There are whole conferences dedicated to this frog. But we can measure methylation using this array. And as an aside, I briefly mentioned we even built a clock for this frog, But what's interesting? Well, we now focus here on cytosines that are so conserved that they are present in all mammals and also in frogs, But there was a challenge, and this is the following. We had, I'm making up a number, let's say we had 100 frog samples. But unfortunately, we lost half of them. We couldn't use them. And that was due to a software issue, not a data quality issue. And now I'm talking to the experts of pre-processing IDAT files, right? If you have an Illumina array, and initially there's an image file, right, a picture. And then there's an algorithm that turns that picture into an IDAT file. And this algorithm does not quite work here for the mammalian array. It needs to be tweaked. So, and this is an open research question. So maybe there's somebody in the audience who really wants to do the world a huge favor, because if you solve that, and it's just threshold choices, right? What's the issue? There's background, you need to change the thresholds, suddenly it works beautifully. So you would do the world a huge favor, because we lose half the arrays, think the monetary loss, but more importantly, the priceless data loss. So somebody can really make a difference. So in particular, we've applied the array even to fish, right? Fish is even more distant than amphibians from mammals. And so yes, I think one could tweak the software so that we can generate data from fish. And there would be something beautiful about it, because the entire methylation aging field, right, this insight that aging affects the methylome, goes straight to this study from a Russian researcher, Berdyshev, who in 1967 actually looked at this particular humpback, sorry, I need to get this, humpback salmon. And he showed that the humpback salmon loses methylation as we age. So the field is old, but it would be pretty cool if we could actually go back to that species and importantly, connect the humpback whale to humans, because it would then look at ultra-conserved stretches of DNA, So yeah, so that was one open research question. And throughout the talk, I will highlight a couple of open research questions. So now I talk about briefly these third-generation clocks. Earlier today, Rosa talked about second-generation clocks, which are mortality risk predictors. I want to talk now about third-generation clocks, which are multi-species clocks. And let's start with primates. So primates are separated by 55 million years of evolution. And you would think, well, maybe it's almost impossible to build a clock, one regression model, one elastic net regression that applies to all of these species when it comes to measuring age. And what I can tell you is the exercise is trivial. Anybody could do it, as long as the data are generated on this mammalian array. Highly conserved cytosines, extremely strong signal. So here I show you a pan-primate clock that applies to 37 primate species, And before I move on, I need to introduce a key concept, which is relative age. Age as opposed to the regular age, which we call sometimes calendar age. So the relative age is simply the age of the animal divided by the maximum lifespan of the species. And as an example, I mention a 2-year-old rat has a relative age of 0.5 because the maximum lifespan of the rat is 4 years. So 2 divided by 4 is 0.5. And in humans, the maximum lifespan is 122. So a 60-year-old human will have a relative age of 0.5. So, and a lot of our multi-species clocks use relative age as a dependent variable, right? So it's same old, same old. You use some sort of a elastic net regression, But the dependent variable, the outcome variable, is relative age. And for example, here I show you a clock that applies both to humans and also to rat tissue samples. The left panel shows you in green the human tissue samples, in orange the rat tissue samples, and on the x-axis you see relative age. And by the way, every scatter plot you see is based on cross-validation, so it's unbiased. But to me, this is already quite remarkable that you can build a clock that applies to rodents and us. So why is it biomedically interesting? Well, because all preclinical studies are based on rats and mice, right? And so having one clock based on these highly conserved cytosines that applies to both species could be helpful to increase the chance of translating various interventions to humans. All right, but so this is a multi-species clock, but Ake Lu, who is here at the conference, presented a poster where she described her pan-mammalian clocks. So Ake built several universal clocks. Here I show you the clock for relative age. The right-hand side shows, again on the x-axis, relative age. But here we're looking at a couple of hundred species, and it's just absolutely remarkable that one can observe such a high correlation. To me, this shows that certain parts of epigenetic aging are very conserved. And here is Ake's universal clock applied to different species. The upper left shows you human samples. Then we have, of course, in the middle, rodents. We have dolphins. We have wolves, elephants, bats. So stunning. And here is a very colorful plot, and I just want to briefly run you through it. You see in the middle a spiral. And the spiral encodes the maximum lifespan of different species. So in other words, the species are arranged by maximum lifespan. And then you see the jaggedy lines on the outside, which indicate the accuracy of the clocks, the universal clocks. And accuracy measured by the correlation, predicted age versus actual age of the animal. And the point is, there is no spiral for the accuracy, right? No pattern. And what does that mean? It means that the accuracy of a universal clock does not depend on the maximum lifespan of the species, And well, to step back, as you know, people have built very accurate clocks for humans, very accurate clocks for mice. Both have very different lifespans. And it's kind of an interesting finding, right, that epigenetic clocks in general, the accuracy of the clocks does not depend on the lifespan. Because one could have thought that, well, maybe you can only build epigenetic clocks in long-lived species, right? But that's not the case. And I dwell on this point because it relates to some of the formulas that I show later. Later we'll talk about invariance. In any event, so we have now these pan-mammalian clocks. And let's just ask some very basic questions. Do universal pan-mammalian clocks predict human mortality risk? And here in the upper panel, I show you Ake's analysis in the Framingham Heart Study, the Women's Health Initiative, in different racial groups. And the answer is yes, they predict human mortality risk weakly, right? I mean, it's clinically not useful, but in an epidemiological sense, it's highly significantly associated with human mortality risk. And so that's cool. And And then there's another nice result for the experts. Some of you know, of course, Yamanaka factors, OSKM, can reprogram cells into iPS cells, and that completely rejuvenates the universal clocks, similar to what it does to all other clocks. Now, why do I show the results? Because you should be asking, well, what data are being used here? The Framingham Heart Study, do you think they ran the mammalian array or the Women's Health Initiative? Of course not, you know. They ran the 450K array. And so what Ake had to do is she had to impute mammalian probes using 450K data. And this brings me to a very important open research question, very generally. How do you translate biomarkers that were based on the mammalian methylation array to biomarkers for human Illumina methylation array data, 450K data, EPIC data, or vice versa, right? Imagine there was a wonderful talk just before mine on methylation estimates of C-reactive protein in humans. Very interesting biomarker. Wouldn't it be cool to evaluate the methylation estimate of C-reactive protein in hundreds of species? Which species has high inflammation, which has low? Cool question. But to implement it, you need to accomplish this translation from one array platform to the next. And so I invite creative people to develop far better imputation methods. And we have data. We generated some data on the EPIC array and the mammalian array. So if you get inspired, that would be very important to solve that problem. But for the people who generate data, the epidemiologists, I urge you to find ways to actually measure the mammalian array in your cohorts, right? Because instead of, for example, I told you in the earlier slide that the universal clock predicts mortality risk, but not really well. But why not well? Well, maybe it's the issue that we used imputation and it's imperfect data, do you see? We lose a lot by going from one platform to the next. So it would be invaluable if one of the large cohort studies could actually measure the mammalian methylation probes in that human cohort study and then relate all these insights from comparative biology to human health. All right, now I want to come to a topic that many of you talked about, epigenome-wide association studies. And Ake found many cytosines that relate positively and negatively to chronologic age in all these mammalian species. And the experts in the room will not be surprised to learn that age-related gain of methylation often happens at bivalent chromatin regions, target sites by polycomb repressive complex 2. So that's known. But can we delve deeper into that? And so we do it, what many of you do, we use chromatin states. And again, chromatin states developed by Jason Ernst's laboratory. It's called universal chromatin states. I personally feel a better name would be pan cell type chromatin states. You know, so these because these chromatin states apply to pretty much to most human cells, and here. I just want to illustrate that. So on the rows are these 100 chromatin states, universal chromatin states. And then the columns are different tissues, but excitingly human tissues and mouse tissues, okay? And what do you see? There's a consistent pattern. If there's a chromatin state that has high methylation in humans, and really meaning in most human cells. It also has high methylation in most mouse cell types, and or conversely low methylation. For example, I will be talking a lot about bivalent promoter regions. It's a chromatin state, and it has low, it's here at the bottom. It has, blue means low. So bivalent promoter regions have low methylation in all tissues and both human and mice. And so, although the chromatin state was originally developed for humans, we apply these chromatin states to all species. And then a careful analysis by Arke on, well, which chromatin states gain methylation, which lose methylation, led to several insights, and I just highlight what I just said. These bivalent promoter regions are highly enriched with cytosines that gain methylation with age. And all right, now we will come to these fundamental equations that I'm trying to describe to you. And I like them because they bring in some mathematics. And for the people who remember calculus, there is this rate of change, right? First derivative. But it's also known as velocity or gradient or whatever, rate of change across the lifespan. And the idea is, well, maybe a species that has a fast rate of change, methylation increases very quickly, maybe that species lives a shorter life, right? And this is an old idea. There are wonderful papers out there from 5 years ago. So it's an old idea, but people didn't have sufficient data to really test it. Now, the Mammalian Methylation Consortium generated data from hundreds of species. We can test that idea very rigorously. And here I show you kind of the results of that idea. So we, because the answer will actually depends very much on the chromatin state. So let me walk you through the graph. So each dot is a different chromatin state. And on the x-axis, we plot the relationship between the rate of change in methylation and 1 over the lifespan, right? And so what does it mean? Well, these bivalent promoter regions, for example, this idea holds water, that rate of change in methylation relates inversely with maximum lifespan. But there are, of course, other regions where you see the opposite behavior. And there are plenty of regions in the DNA where rate of change in methylation really has no relationship whatsoever with lifespan, you know. So one needs to be careful about how you select cytosines. That's really critical. But if you do it well, or if we focus on these bivalent promoter regions, and then we plot the rate of change, AROCM stands for average rate of change in methylation in bivalent promoters. If we plot it on a log scale, we do see this remarkable correlation between the rate and 1 over lifespan. All right. However, let's be careful. When you calculate a velocity, a rate of change, then and you average over the entire age range, you're glossing over a detail, which is that initially there's a non-linear relationship, you know. So the rate of change often becomes linear roughly after age at sexual maturity, you know. Even in humans, after age 20, it becomes pretty linear. But early in life, there's non-linearities. And so the easy way to, by the way, this is pig blood, but I could have shown you many other species and tissues where we observe that. And so one can define a rate of change in young animals, this is the green, or a rate of change in old animals. That's the blue line. And what you see is the rate of change in young animals is high, and in old animals lower. And this insight generalizes to most species. So here I show you the rate of change in young animals. Each dot is a different species. And on the y-axis, the rate of change in old animals. And the first thing you observe is lower, right? Young animals, fast rate. But there's a second very interesting insight. There's actually a pretty strong correlation. You know, the correlation is 0.82. So to me, this is interesting. And it relates to a talk by another speaker linking developmental processes, or prenatal development, but also postnatal development, right? Rate of change before adulthood to rate of change later, you know. So there's this general rule that there is a relationship. In other words, species that develop quickly, they also show faster aging patterns, you know. All right, and so here I show you the same in different species, the different patterns. Yeah. And let's see. Now I actually go to math formulas, you know. So now is the time to urgently get a cup of coffee for the people. But for the rest of you, stay on. So why math? Because everything I've shown you so far, any of you could have done, but there is a major pitfall, and people in the community are not aware of it, you know. So I see paper after paper where the authors step into a bias, you know. And to reveal the bias, we need a bit of math. All right. So I kept talking about rate of change. Well, how would you actually define it? Well, you have methylation and you have an age, right? And then from calculus, you know the slope, right? Rate of change equals the slope. So this coefficient, that's the rate of change. So far, so good. If you go to your biostats class or statistics class, for this coefficient, there actually is a formula. And the formula, it relates here's the Pearson correlation between methylation and age. Here is the standard deviation of relative age. Do you remember relative age is age divided by lifespan? And then here you have lifespan, you know. And so this math equation didn't use any data. This is simply math. And it should be deeply alarming to you because remember I showed you all these graphs, rate of change relates to lifespan. If you see this equation, you say, but why did you even use data? This is trivial. There's a direct relationship, you know. Do you get that? Yeah. It's, to me, this is deeply disconcerting, you know, because it's just baked into the definition of rate of change that there's an inverse relationship with 1 over lifespan, you know. And most people are not aware of it. And why do I say, what do I mean by most people? There are people that show telomere shortening in different species relates to 1 over lifespan. Then there are papers, beautiful papers, rate of change in somatic mutations relates to 1 over lifespan. DNA repair, mitochondria, 100 things relate to, there are hundreds of articles perhaps on how a rate of change relates to maximum lifespan. But I'm telling you there's a potential bias in the math. And so so let's now go into one question. Well, does the correlation between methylation and age, right? Methylation is just methylation in the bivalent promoter regions. Does that Pearson correlation relate to lifespan? And here I show you Pearson correlation versus lifespan. Long story short, there is no relation. It doesn't, you know, which is interesting. Remember earlier today I told you how universal clocks are equally accurate in short-lived animals as in long-lived animals. To me, this has a lot to do with this finding, that the correlation between methylation and age does not depend on lifespan. All right. Let's go back to this formula. This standard deviation of relative age, what does that mean? So what would be the perfect study? Well, if the mammalian methylation consortium had collected animals in a uniform fashion across the age range, right? From age 0 to maximum age, uniform distribution. But that's not what happened, right? For some species, we had mainly young animals, for other species, mainly old animals, and just different distributions. And all of these distributions have then different standard deviations of relative age. So what I was talking about, the rate of change in methylation, very much depends on that. And this is, there's a dependence. And this brings me to this plot. I think most statisticians are not aware of it. I certainly wasn't, and I'm a card-carrying professor of biostats. But when you have a correlation coefficient, let's say between methylation and age, then there is usually actually a positive correlation to the standard deviation of age. There is a bias here, do you see? And that, of course, impedes a lot of the studies I just described. So do this in your own data sets. For example, some of you do meta-analyses, right? You have 10 different cohorts. Why don't you do a correlation of methylation of age with age versus standard deviation in the 10 different cohorts? You will see this kind of pattern. It's unattractive. And therefore, we defined adjustments of these rates of change. So we define an adjusted rate by multiplying it by a power of the standard deviation. So anyways, I won't go into it. I think you're plenty traumatized. But let's now go here. So when we use the adjusted rate of change of methylation versus 1 over lifespan, we see a correlation of 0.96, do you see? And this is partially due because we corrected for imperfect study design, maybe I come to then a summary. So what did we learn? So yes, the average rate of change in methylation in these bivalent regions has this beautiful relationship of 1 over maximum lifespan. And the second is that correlation of methylation with age bears no relationship to maximum lifespan. You could call it an invariant. And the third fundamental equation is that the young animal's rate of change is proportional to the old animal's rate of change. These are what I call the 3 fundamental equations of mammalian methylation studies. Now I want to come to an open research question. So here I show you the mammalian methylation data, and here we show methylation in bivalent promoter regions, and here's relative age. And I see roughly this black curve, But it would be pretty cool if somebody actually analyzed this very rigorously. So describe this nonlinear relationship between methylation and relative age in different species. And why is that interesting? Well, because Rosa earlier described how she studies developmental processes, right, in pediatric cohorts. You get a rate of change, But wouldn't it be nice if we really understood the non-linearities, And when you do this for individual cytosines, it's exceedingly difficult. But if you average, right, if you average over hundreds of cytosines, then you reduce the noise, such as here, and you see better patterns, you know. So to get you started, here's the best relationship for this nonlinear behavior that Zhe Fei and my team came up with. And, but hopefully you can find a much more accurate formula, you know. Let me now come to a slightly different topic. So we are done with rate of change. Let's do another analysis. We have 348 species. For every species, I know the maximum lifespan. Why don't I form some sort of a regression model? Why don't I form a predictor of maximum lifespan? And this work was done by Caesar Lee, and he developed a pretty accurate predictor of maximum lifespan of a species. So what it means is you go in a holiday, you travel to some jungle, you find some roadkill, you cut off a piece of the tail, you bring it to me, and you say, I don't know what species it was, but can you tell me what's the maximum lifespan, you know? So, and you don't even need to tell me what the tissue type is. I mean, we would be able to tell you roughly the maximum lifespan. But similarly, we have far more accurate predictors for gestation time, okay? Maximum lifespan is a noisy outcome. Gestation time has been measured much more precisely, right? And so for gestation, we can tell you gestation time, we can tell you the average age at sexual maturity, you know. So these species characteristics, you know. And so on some level, you could call this a biomarker. We have of a species characteristic. And if you have a biomarker, the people who do epidemiological studies, they would say, well, can I use this predicted maximum lifespan and relate it to age, you know? And here I show you that in some species, we do see an interesting pattern. So in humans, there is actually a slight relationship with age. But then in other species, we see the opposite. We see negative relationships with age. And do you see what I would have expected? That there's no relationship with age, right? Because why is that? The maximum lifespan of a species is really set early on in development. So whether you predict it based on a child or based on an 80-year-old, in my worldview, the species lifespan shouldn't change. And I do want to remind you, the oldest woman who ever lived was a smoker, you know. And so here, for example, I show you also data from Arke, where she related this maximum lifespan predictor to human mortality risk, no association. She related it also to smoking status, smoking pack years, no relationship, you know. So it's kind of interesting that this species characteristic, the human species characteristic, doesn't relate to traditional risk factors of mortality risk. But this brings me to this earlier research question, because again, when Arke did this analysis where she applied the maximum lifespan predictor to human cohorts, she used some imputation scheme. But I just feel it would be very exciting to revisit these questions with better imputation schemes and ideally with data that were actually generated in human cohort studies. So then, overall, I was disappointed about these results because wouldn't it be nice to have an intervention that does increase our maximum lifespan? I mean, and there's some good news because we did come up with a way. And this is the following. we looked for sex effects in different species, and we found 18 species where there was a significant difference between predicted maximum lifespan in males versus females. And one of the most significant results was humans, human samples. And it's kind of trivial. We all know women live slightly longer than men. But there's my intervention for you, you know. If you're a man, there's some hope for you. Just turn into a woman. And so, now I want to talk about EWOS of maximum lifespan. So we can, of course, also look at cytosines that correlate with maximum lifespan. And the experts in the room will know, well, you need to adjust for phylogenetic relations. There's something called phylogenetic regression. You need to possibly adjust for adult weight, right? Bigger animals live longer, could be a confounder. So there are many confounders. And what I want to tell you, we did all of the above. We adjusted in a million different ways. So I invite you to read the paper. But let's just go with the analysis here. What were the insights? One was that, and this is this Venn diagram. So cytosines that relate to maximum lifespan typically differ from those that relate to chronologic aging effects. In short, it's quite different. And it comes to this point that I made, you know, that overall maximum lifespan is set at birth, so to speak, you know, and it doesn't relate to chronologic aging effects in most locations. And okay, so you have EWOS, and you can do some grade enrichment or functional enrichment studies. And what do we find? Overall, we find developmental processes, which makes sense. In particular, Hox genes, you know, interestingly, body patterning genes. So really development. Reinforcing what I said, somehow maximum lifespan is really set very early in development. But there was another interesting finding, as one can do upstream regulator analysis, in certain ways find exciting transcription factors that could explain methylation changes associated with lifespan. And that implied OCT4, NANOC, so these SOX2, so these pluripotency factors, these Yamanaka factors, they were also implicated when it comes to maximum lifespan, interestingly. And I want to describe one of the key insights about long-lived species, which is the but let's start slowly. As most of you know, we do observe low methylation in transcriptional start sites. And so the blue line is the line mean methylation in species. And conversely, we often see higher methylation in the gene body, or also high methylation in intergenic regions. And now that's what one expects from our many human studies and also mouse studies. The red curve shows you, in certain ways, the association with maximum lifespan. And what the finding says is that long-lived species have high methylation or maintain high methylation in regions where you want to have high methylation. They maintain or low methylation in regions where you want to have low methylation, you know. To say this, well-crafted methylation landscapes are really a hallmark of long-lived species, you know. A long-lived species will have high mountains of methylation and deep valleys of methylation already at birth, you know. Whereas short-lived species have a more uniform landscape, a flatter landscape. Cells develop quickly, mature quickly, and the methylation landscape is noisier, is flatter. I want to slowly end. I started a non-profit foundation that sells the mammalian methylation array. It's fully available to anybody, free open science. All of our software is free. And so what I told you about today is you can build pan-mammalian clocks. You can estimate maximum lifespan with cytosine methylation. We have epigenetic clocks for all model organisms. And above all, I invite you to delve into our publicly available methylation data. Hopefully you find them useful in your research. And I stop with the acknowledgement. Thank you.
Thank you very much, Steve, for a great overview on your recent work. Dragging it on the floor. No one can hear me. Thank you for a great overview of your recent work. I'm going to kick off with one question, or people are thinking questions. And there's a kind of puzzle I always think about with these clocks, because they always start at day 0, which is obviously birth. But obviously, life actually really begins, sort of, in humans, 90-odd, 40-odd weeks before that. And we know, certainly that there's lots of crazy changes that take place at that time, and obviously different species have different gestation periods, as you alluded to. So how did you factor that into these clocks? Because, for instance, mice are born equivalent of sort of second trimester. In human, we're in utero for much longer. So I'm just intrigued how that kind of fits into this, because that would be, to me, a critical possible limitation.
Yeah, well, when you go through the methods section of our paper, you will notice we do account for gestation time. We do, it's a parameter. However, I would turn it around and say one could revisit what we have done. For us, gestation time was a nuisance parameter. We dealt with it in order to avoid negative numbers. So in our database, negative always means prenatal, samples. And above all, we want to avoid taking a logarithm of 0. So anyway, or negative numbers. So we dealt with it. But in theory, there's an opportunity to build these pan-mammalian clocks that really hone in on developmental processes, because we were more interested in postnatal processes. And earlier today, some people mentioned these clocks that measure gestation time, like cord blood-based clocks, or even placental clocks can. There's an opportunity to build these pan-mammalian clocks for early development. We have a few prenatal samples in our database, I should say, but not many.
Okay, well, we should talk after. We have a lot of fetal samples. It'd be really interesting to maybe run the clock on those. Anyway,
yes.
Into the audience. If everyone puts it, it's quite blinding here, so you have to really wave your hands to get attention.
Thank you, Steve. Very interesting talk. So I guess my question relates to this observation that there are already sequence features, so the mammalian clock and maximum lifespan. So there are already sequence features between mammals, right? And we know that to a certain extent those are predictive of maximum lifespan. To what extent do you think, obviously the methylation predictor does much better, but to what extent do you think that might be genetically encoded? And to what extent is that some non-genetic influence?
Yeah, that's a very important question. It depends a bit on the particular analysis one conducts, So for some of the claims that we have, let's say we have a cytosine, and we say the methylation of that cytosine relates to maximum lifespan. Then there's a concern that this simply encodes a sequence difference, some sort of a mutation, And it is a concern. We addressed it by kind of carefully looking at it, you know. So we, for example, you can hone in on subsets of species where there's literally no mutation in the vicinity of the particular cytosine. So you de-risk it, you know. Another question is, well, I presented, for example, this the multivariate model to predict maximum lifespan. And then one would say, do you really have to measure methylation? Can't you use phylogenetic relations or sequence properties, you know? And so we very carefully compared these two types of predictors, basically one that looks at phylogenetic information and one that uses methylation, to answer the question, is it worth the money to measure methylation? And as you indicated, there was a benefit, you know, so methylation adds some information, but conversely, clearly phylogenetic relations very much encode also lifespan, you know. Any species that's closely related to a mouse, most rodents are short-lived, most primates are long-lived, you know, so no doubt it's part of that. Yeah. As all of you know, methylation is very much under genetic control, you know. So it depends really on the analysis, you know.
I saw a vigorous hand over that
Yeah.
way.
Yeah. Hi, thanks. Really lovely talk. So my question wants to probe you a bit more on what you think the mechanisms underlying both rate of change of methylation at clock CPGs and lifespan CPGs are. I know there's been some work, I think you were involved as well in the work that had the Down syndrome mouse with the human chromosome 21, and that shows in the mouse that chromosome actually ages at the mouse rate, not at the human rate. And then also, I think what's interesting in these heterochronic parabiosis experiments, you can rejuvenate basically an animal by linking its bloodstream with a younger animal, right? So that suggests it's something in the environment as well. It's not just purely encoded in the genome of the cell that is aging. So what do you think it is? And is it different for lifespan and for clock?
Yeah, you're asking the critical questions. Thank you. When it comes to this very interesting study where Rob Lowe and I think Bartman, Rakin, and they showed that this Down syndrome chromosome methylates faster in a mouse. It's fantastic work, you know. But I was not involved. I wish I was, you know. But yeah, honestly, I have no idea. I mean, there's something, I wish, zero idea, you know. What I know is there's, of course, a daily turnover in transcription factors, you know, and that this metabolic rate is also quite different, you know. And so it's an obvious idea that somehow that plays a role. But if we forget aging and everything, if you just ask the question, developmental speeds, why does a mouse have a gestation time of 21 days, you know? And there's so much work on that question, what controls developmental rate. And I would suspect many of the answers will also relate to human aging. And the reason is this one law I showed you, right? Whatever, when you have a species with fast rate of change in young, it also has fast rate of change in old, you know. And so I hope that these insights from developmental biology will also be useful for aging studies. Yeah. But I don't have a good answer. Yeah.
I think Bas has some online questions.
From Art Petronis. Great talk.
Thank you.
Did you include a naked mole rat?
Yes.
Nice, because then these are very long-lived. Any insights there? Any anomalies?
Yes. So our study wanted to collect very long-lived species, for sure. Naked mole rat, bats. Let's not forget humans, it's very long-lived. A bowhead whale. We have multiple organs from the bowhead whale who lives 211 years according to the Animal Age database. But conversely, I was very interested in utterly short-lived species because mice are not the shortest lived. They're shrews, you know, that are much shorter lived. And anyways, so what are the insights? the naked mole rat, from an epigenetic point of view, is completely boring as humans, you know. Because, so my team published a naked mole rat clock, Vadim Gladyshev in parallel. So there are now several epigenetic clocks for naked mole rats. They work very accurately. And in hindsight, it's all obvious. Naked mole rat is long-lived species. Humans are long-lived species. Why shouldn't we be able to build it, you know? But then a naked mole rat is a famous animal because people claim it has negligible senescence. And that may be true or may not be true on some phenotypic readouts, but I can assure you in terms of methylation, there's no negligible senescence. And we were hoping to find one organ that is special. So we said, oh, maybe the blood is different, or the liver, or the brain. And in all naked mole rat organs did we see methylation aging effects. All of our data are publicly available. Yeah. Thanks.
There's a question there.
Yeah, hi, I'm Eric from the University of British Columbia, and I was interested in the observation that many CPG sites that are associated with age display a nonlinear behavior. And I was wondering if there's any reason of why most of the clocks are trained using ElasticNet, which are like regularized linear models, which might be related to some clocks predicting well in the intermediate spans of lifespan, but not in the extremes. So yeah, the question is there any reason of why ElasticNet is the mostly used algorithm, or should people move for models that better model nonlinear relationships such as trees?
Yeah, I do think it would be exciting to use nonlinear models. But just to be very clear, remember that we often, in 90% of all cases, we apply a nonlinear transformation to the age variable. So the dependent variable undergoes a nonlinear transformation. But you could ask, well, should the covariates be entered in a nonlinear way? You know, for example, a neural network or some spline, or, you know. And yeah, I encourage people to look at it. It could probably allow you to build sparser clocks, you know. So our clocks, a universal clock may use 600 cytosines. So if you more carefully think about it, maybe you can select fewer cytosines, you know. So long story short, it's an exciting exercise, but you need very clean data. I think that has hampered the field, not small sample sizes, you know, leading to false positives.
I think we've got one more question. Chris.
Thank you. Very thought-provoking talk, as always. I was just wondering, in terms of the enrichment that you find with bivalent domains, if you've sort of delved into that any further, because I know in that universal chromatin segmentation, there's actually sort of 3 subclasses that they categorize those as. Was there a particular enrichment for a particular subclass of bivalent domain, or was there some particular part of the signal that's being picked up there that's more enriched?
Long story short, we haven't delved deeper into that, you know. So these 100 chromatin states, as you said, there are several bivalent promoter regions, bivalent promoter 1, 2, 3, 4. And number 2 is particularly enriched, you know. And but one could delve much more deeply. What I want to say is you need to think also about tissue type. The enrichment is stronger in proliferative tissues, you know. And overall, I think it's very worthwhile to distinguish nervous tissue from peripheral tissue in everything we do, you know. But yeah, the data are publicly available. So what I learned when I listened to your talks, I always said, oh my God, they are so much better at EWAS than I am, you know, because if you saw my EWAS, you wouldn't let it go through peer review, you know, because you use these very sophisticated tools. So please revisit our EWAS, do it better, use better annotations, you know, move things forward. There's really an opportunity here.
Thank you.
Well, I think we've run out of time.